Now, three of the amino acids which are shown here, that is glycine, alanine, and serine. You can see here the R group. The R group here has been changed in all the three. A hydrogen amino acid in the glycine, and a methyl group in the alanine, and a hydroxymethyl group in the serine. So the chemical and physical properties of amino acids, of the amino, carboxyl, or R amino, carboxyl, or R, that is the functional group, that is glutamic acid, lysine, or any amino acid, depends on these functional groups. So there are aromatic amino acids also, which are lysine, valine, and there are many aromatic are tyrosine and phenylalanine. So the property of the amino acids, they are because of their ionizable nature of the amino group as well as the carboxyl group. So in different pH, the different structure of amino acids changes. Lipids are generally water, they are water insoluble. They can be simple fatty acids which have the carboxyl group attached to it. They can be simple fatty acids like carboxyl group attached to an R group, or the R group could be methyl group or ethyl group. It can be ethyl group, methyl group, or higher number of methyl groups also, like 1 to 19 carbon atoms they can be there. Like here it is palmitic acid. It has 16 carbon compounds in it, including carboxyl carbon. Okay, so fatty acids, they could be saturated, that is without double bond, or unsaturated with one or more double bonds, they are present there. So we find that a simple lipid is the glycerol. A very simple lipid is the glycerol. It is a trihydroxypropane. You can see here tri, 3 carbon atoms, hydroxyl groups are there. So it is also called as trihydroxypropane. So many lipids have both glycerols as well as fatty acids. Both are present in them when they become more complex. So both glycerol and fatty acids could be present in some of the lipids, and the fatty acids, they are found esterified with the glycerol. Okay, when fatty acid is esterified with the glycerol, means an ester bond is formed in between. Then they can be monoglycerides, diglycerides, or triglycerides. They can be monoglycerides, 1 group, diglycerides, 2 groups, or triglycerides with 3 groups. They are present in the fatty acids. So these are also called as fats and oils based on their melting points. Oils, they have low melting point, like any oil, you can say gingerly oil, and they remain as oil in winters. So some lipids, they have phosphorus and phosphorylated organic compounds in them, and these are called as phospholipids. You can see here. Now these are phospholipids because of the presence of phosphorylated organic compounds or phosphorus in them. They are found in the cell membranes. Then lecithin. Lecithin here, this is a phospholipid. So lecithin is one such example, which is found in the cell membrane. Some tissues, especially neurons, neural tissues, they have lipids with the more complex structures. So living organisms have a number of carbon compounds in which heterocyclic rings they can be found. And some of these are nitrogenous bases: adenine, guanine, cytosine, uracil, thymine. And then when they are found attached to a sugar, so you can see these heterocyclic rings, and when they are found attached to a sugar, then they become, when they are found attached to the sugar, they are called as nucleosides. You can see the structure here, nucleosides. And if a phosphate group is also found as esterified to the sugar, then they are called as nucleotides. You can see here the phosphate group is attached to the sugar. These are the nitrogenous bases: adenine, uracil, Okay, these are purines and these are pyrimidines. So if a phosphate group is also found attached to a sugar, they are called as nucleotides. So if a phosphate group is also found as esterified to the sugar, they are called as nucleotides. And adenosine, guanine, cytosine, thymine, they all are nucleosides. So adenylate acid, thyminic acid, and ganglionic acid, uralic acid, and cytolytic acid, these are all nucleotides. So nucleic acids like DNA and RNA consist of ribonucleotides, nucleotides only. So DNA and RNA, they also function as the genetic materials. Primary and secondary metabolites. Now, when we are dealing with chemistry, we deal with isolating thousands of compounds, which can be small, which can be big, from the living organisms, and we determine their structure by we synthesize them also. So if we have to make a list of biomolecules, there are thousands of organic compounds which are amino acids, sugars, and they are given here also in this table. We can call these biomolecules as biometabolites. So animal tissues, we see the presence of all such categories of compounds. All these are there. They are called as primary metabolites. But when we analyze plants, fungus, or microbes, we see thousands of compounds which are other than these primary metabolites are also there. These can be alkaloids, flavonoids, rubber, essential oil, antibiotics, colored pigments, scents, gums, spices, etc. These are called as secondary metabolites. So primary metabolites, they can function and play their role in the normal physiological processes. Now we can also understand the role of the secondary metabolites in the host organisms. Many of them, they are useful to human beings, like rubber, drugs, spices, scents, pigments, etc. Some secondary metabolites have their ecological importance also. Now this is the list of some of the metabolites which are primary as well as secondary metabolites. These can be alkaloids, terpenoids, essential oils, toxins, lecithins, drugs, polymeric substances, pigments, etc. Then carotenoids, anthocyanins, etc. These are the pigments. Alkaloids like morphine, codenine. Then terpenoids like monoterpenes and diterpenes. Essential oils, lemongrass oil, abrin and ricin, these are the toxins. And concanavalin A, these are the lecithins. And there are many such other secondary metabolites. Biomacromolecules. Now there's one feature which is common to all the compounds which are found in the acid soluble pool. They have a molecular weight which is from 18 to 800 Daltons, and the acid insoluble fraction, it has 4 types of organic compounds. These are proteins, nucleic acids, these can be polysaccharides, and lipids. So these classes of compounds, with the exception of lipids, they have a molecular weight in the range of 10,000 Daltons and above. So for this reason, biomolecules, that is chemical compounds which are found in the living organisms, they are of 2 types. One which have a molecular weight which is less than 1000 Dalton, and they are referred to as micromolecules or simply biomolecules. While those which are found in the acid insoluble fraction, they are called as macromolecules or biomacromolecules. So the molecules in the insoluble fraction, with the exception of lipids, are polymeric substances, polymers, okay, which are made from many monomer units. So then why do lipids, whose molecular weight do not exceed 800 Daltons, come under acid insoluble fraction, that is macromolecular fraction? So lipids are very small molecular weight compounds which are present not only as such but also arranged into structures like cell structures and other membranes. So cell membrane and other membranes, they can be broken into pieces. They form vesicles which are not water soluble. Therefore, these membrane fragments, they turn into vesicles, they get separated from the acid insoluble pool, and thus in the macromolecular fractions, lipids are not strictly macromolecules. So the acid soluble pool, which represents roughly the cytoplasmic composition, the macromolecules from cytoplasm, organelles, they are acid insoluble fractions, and they represent the entire chemical composition of the living organisms. So the chemical composition of the living tissues from abundance point of view, large point of view, if we arrange them class wise, then these are all in the form of these. Just a second, the previous slide, these are all in the form of these previous slide that we have seen. These are in the form of these only: water, proteins, carbohydrates, lipids, and nucleic acids and ions, with an exception of lipids. Proteins. When we come to proteins, they are polypeptides. Polypeptide means many peptide chains are together form in the protein. They are linear chains of amino acids which are linked by peptide bond. Now, each protein is a polymer of amino acids. There are 20 amino acids: alanine, cysteine, proline, tryptophan, etc. And a protein is a heteropolymer and not a homopolymer. Homo means same type of units, and hetero means different types of units may be attached. So homopolymer has only one type of monomer which is repeating many number of times. So we also find that amino acids, they can be essential or non-essential. So non-essential, which our body can make, while we get essential amino acids through our diet or food. So proteins, they carry out many functions in the living organisms. Some they transport, others some fight infections, some in the form of hormones, enzymes. Collagen is the most abundant protein in the animal world, and ribulose biphosphate carboxylase oxygenase, which is also called as rubisco. It is the most abundant protein in the whole of the biosphere. So that you have to remember, rubisco, which is present in plants. It's a collagen form. It's in the form of a protein, and it is the most abundant protein in the biosphere. So apart from this, various other types of proteins, they are mentioned here and their functions. Polysaccharides. The acid insoluble part, it has polysaccharides. They are also called as carbohydrates. It's another class of macromolecules. So polysaccharides, they are long chain of sugars. You can see here, these are the chains which are attached to one another. These are the long chains of sugars. There are threads, just like cotton threads, containing different monosaccharide units. So these are the monosaccharide units. And cellulose is a polymer, which is a polysaccharide, which consists of only one type of monosaccharide, that is glucose. And glycogen is another animals, they have glycogen. And insulin is a polymer of the fructose. And polysaccharide chains, say glycogen, here the right end is called as the reducing end and the left end is called as the non-reducing end. So you can see here they are in the form of chains and the branches. So starch, it forms a helical secondary structure. Starch can hold various molecules in the helical portion like this. And the starch, iodine, it is blue in color. So cellulose does not contain complex helices, and that is why it cannot hold iodine, whereas starch has helicals like this, so it can hold iodine together and form the blue-black color, whereas other cellulose, it cannot do that. So plant cells, they are made up of cellulose. Paper, which is made from plant pulp, as also contain a fiber as cellulose. And there are many complex polysaccharides in nature. These are the building blocks. These are amino sugars.